with that, I'm going to introduce our speaker today. So we are really excited to introduce Dr. Anne-Marie Baird, one of our STARS mentors. She is an experienced cancer molecular biologist and currently a senior research fellow in Trinity College, Dublin, Ireland. She became involved in lung cancer patient advocacy in 2012, starting with her participation in the lung cancer social media, the hashtag LCSM community on Twitter. And she's one of the founding members of the Irish Lung Cancer Community, ILCC, and she's the current president of Lung Cancer Europe. So we are really excited to welcome her today. Please give her a warm welcome. And with that, I will pass it over to Anne-Marie to start the presentation.
Thanks so much, Sarah, and it's lovely to be back to give this presentation. So I should say that I may not be able to see everybody while I'm giving this, but I know Sarah said questions will be at the end anyway. So it's great to be here to give this talk again this year, which is just going to be a bit of a brief introduction to the science of lung cancer. And before I start, just thanks to all the partners and supporters of the STARS 2024 program. So a brief overview of what I'm going to cover, because I know we want to leave lots of times for all the fantastic questions everyone submitted. We're going to talk a little bit about getting back to basics. We're also going to touch upon where we are now, but obviously in the interest of time, we can't give an exhaustive overview of everything. And then what might be coming next in the lung cancer space. So when we think about giving back to basics, the primary function of the lungs is to deliver oxygen to our red blood cells and to remove carbon dioxide at the most basic level. That's the primary function. So it all sounds like it should be a very sort of, I guess, simple organ with this one main function that it would carry out. But of course, we know our lungs are very complex. Not only are they complex given their function, but also in terms of the number of cell types that they contain. So here you can also see that there's a number of different lobes within the lung, and then also the different compartments that are there within the lung, whether we're talking about the large bronchus, the peripheral compartment, or the respiratory bronchiole as well. And you can see that there's quite a large number of different cell types within these compartments, such as a ciliated cell, a mucus cell, and all of these have their own specific function to keep the lungs happy and healthy when everything is going well. But of course, we know that when things go wrong, lung cancer can start in any of these cells within the lung. So given that we see how complex it is in terms of the different cells and how they have to work well together, each cell has to know the job that they have to do, the task that they have to carry out. So things have to be very controlled within the cells. And how that's controlled is really down to cell signaling. So when we think about cell signaling, it's what happens within the cell and it impacts how genes are expressed. So to give that in a little bit of a kind of more visual example, the life cycle of the butterfly, because I think it's something we can all easily imagine in our heads. So if we came from a completely different planet and we didn't know that a caterpillar was related to a butterfly, we might think that we're dealing with two completely different organisms that weren't related at all. But we know, of course, they are with the life cycle of the butterfly. So when you think about it, irrespective of the stage of the life cycle that we're at within the butterfly, the same DNA is contained within the cells, irrespective of whether we're at a caterpillar or a butterfly stage. The difference is that which genes are switched on or turned off at the various cycles of this life cycle is then going to influence where we're at within that cycle. So this shows you just how tightly controlled the cell signaling needs to be and when genes should be switched on and switched off, and our bodies are no different than this. So if we look at that in a slightly different way, we know that cancer can alter the normal cell signaling. So this very tightly controlled process we have in our cells, it can go awry. But initially, if we look at a normal cell, and that cell gets a signal to divide. So what happens is it goes through sort of checkpoint sort of controls when it's doing this. So a little bit like a traffic light system. Are things okay? Are they not okay? Are you going to get the green light to proceed? And what happens within our cells, there's a number of different checks the cell goes through, and they can get the green light to divide into two daughter cells. Sometimes there's damage or there's an issue with the cells, and we have a lot of things that can come along to help repair whatever that damage might be. So you kind of get this amber signal that's a little bit of a pause. Let's see what's wrong and can we fix it? If things get fixed, then the signal is then that they can continue to divide to two daughter cells. But in some instances, there might be a sense, damage might be sensed that can just not be repaired at all. And in this point, when everything's working as it should, that cell will be given a signal that it should die. So therefore, it's not allowed to divide because we're trying to keep everything in balance and as it should be. However, in a cancer cell, when you think of this traffic light system, it doesn't matter what might be wrong within the cell or what behavior it's displaying, but there's this green signal to go all of the time. And that signal to divide all of the time, irrespective of what's happening, is then obviously going to have those cells that are going to form into a tumor. So that's when things are not going as they should. So when these things go wrong, it indicates that something can be of an issue within our DNA. And the simplest way that I look at this is I think most of us are aware of sort of flat pack furniture and the instructions that come with it. So you have to follow it. And when things go wrong, you know yourself that you may not end up what you thought you were going to end up with. So for instance, if you get a flat pack piece of furniture and it's a chest of drawers, and if you obey all the instructions, that's what you get. But if something goes wrong with the instructions, you might end up with a table or a bookshelf, which is obviously not serving the function that you wanted. And this is something similar that can happen within our DNA. Something isn't translated properly from our blueprint, and we end up with something that could be doing a job that's going to initiate cancer and cause cancer cells to continue to proliferate. When we look at that in a more sciencey way and move away from flat pack furniture and IKEA, it's as if it's the same as our DNA, which is our blueprint. Our genes are transcribed into this message. From the messenger RNA, that message is then translated into protein. So for instance, in this case, it would be that chest of drawers that we see there. And then it's the protein that is the workhorse of the cell and does a lot of the different functions that we need for our cells to do. So changes in the DNA can cause cancer. So if we think of that analogy and continue with it, here we have an example where there's a specific gene in the body. I've just referred to it as gene X. There's a signal that gene X needs to be transcribed and translated into a protein. So you go, and this is a very simplistic overview, I should say, from the gene to the message to the protein, and everything as it should be. And you end up with that chest of drawers. However, if something happens within that gene, there's an insult of some kind and a mutation occurs within the gene, as indicated by the little red box here. You can see that that then goes from the gene to the message to the protein. And then you've got a defunct cell here that's carrying that incorrect protein. So then the function is going to be different. So then in that instance, instead of ending up with your chest of drawers, you end up with that bookshelf. But of course, we know as we age, we accumulate different mutations, but not all changes that happen in our DNA leads to cancer. So there's two very important genes that we need to think about when we talk about cancer. And one is termed proto-oncogene, and another is called a tumor suppressor gene. So proto-oncogene is something that's promoting cell cycle, promoting cell division. So if you think back that one cell going to two cells, it's promoting that. So if something goes wrong with a proto-oncogene, usually it means it's switched on all of the time, and then you get that green light all of the time for that cell to continue to divide. But if we think of a tumor suppressor gene, a tumor suppressor's gene's function is to restrict cellular growth and cell division. And therefore, when that's knocked out, it tends to, or when something happens within those genes, it tends to stop that gene from functioning. So those restrictive signals are no longer received by the cell. And then again, you get those cells misbehaving. So an example of a proto-oncogene would be something like EGFR, and an example of a tumor suppressor gene would be something like TP53, which is often also referred to as the guardian of the genome. So changes, that happen within the cell changes the cell signaling, and this change in cell signaling can cause cells to behave like a cancer. And we know that these changes can take place over a period of time. But I just wanted to give you kind of an example of just how complex this is. And here is a diagram for EGFR pathway. So you can see the EGF binds here onto the EGFR receptor, and that causes all of these cell signals to happen. And you can see that there's many different sort of signaling pathways that can be activated. And basically those go in here into the genes within our DNA, and they can cause certain genes to be on or off depending on what the situation is. So it's going to influence what genes are switched on and what genes are switched off. And then if that's incorrect, it obviously is going to mean our cells are misbehaving and behaving like cancer. So when all of these things happen, we have then what's called the hallmarks of cancer. So there's huge studies that have gone into this to show what differences are occurring in cancer cells versus normal cells. And I should say that all of the research that's ongoing, there's different hallmarks being added to this list. It started out much smaller than this, and now you can see that it's expanding. So some of these examples are things like sustaining that proliferative signal, so that signal to keep dividing, evading growth suppressors, so evading those sort of restrictive messaging that it's getting, that cells are getting, that they, shouldn't divide or continue. Then you have other things like genome instability and mutation, activating invasion and metastasis, and so on. But there's quite a large number of different hallmarks that have now been identified within cancer cells. So if we think a little bit about an example within lung adenocarcinoma, I just wanted to show this example of kind of when things can go a bit awry. And this example comes from a paper by Charlie Swanton's team, which was a huge, big study that was published, I think about a year ago, if I'm correct, or maybe in the last 2 years. And it was looking at lung adenocarcinoma promotion by air pollutants. And you can see there's two different models that's shown within this diagram. So there's a model A, and then this newer sort of model discovered by the Swanton group. So if we think within a lung cell, and the lung cell is exposed to different things within the air, in this case, something that could cause a mutation, so a mutagen in air pollution, that's your lung cells are getting exposed to that. And then you end up developing, for instance, a mutation that's induced by this air pollution that your lung cells have been dealing with. Then over the course of time, you can go from a more benign tumor with added insults, so added mutagens in the air pollution, to a tumor that has invasive properties and is fully malignant and is able to metastasize to different sites. So in the Swanton group, what they found is that they have shown evidence to provide a model that there's particles and air pollution that may act to promote the proliferation of lung cancers, or lung cells, I should say, that carry pre-existing mutations. And these pre-existing mutations could be spontaneous mutations that are already there within the cell. So here we have a particular type of cell, particular type of lung cell, I should say, and it has within it a spontaneous mutation, and usually it's an EGFR or KRAS from the study of this wanton group, and that's in a sense lying dormant. So it's there, but it's not causing any issues or harm, so to speak. But then, given the exposure to air pollution and the particles within air pollution, you're getting this inflammation that's then happening around these cells. So, with prolonged inflammation, you are then promoting the tumor forming capacity of these lung cells. So, it's kind of the spark that wakens up these dormant cells. And then you end up with a tumor with invasive properties that acquires additional mutations from the genomic instability that's caused by all of the inflammation here. And then you're getting a fully malignant cancer that can spread to distant sites. So talking about cancer spread, we're talking about metastasis. So cancer can spread via the blood, the circulatory system. So here in this example, you can see we have tumor cells surrounded by normal cells. A cancer cell breaks through this barrier here, so it detaches off from the primary tumor and it breaks through the barrier, and it's here then, and it's going to go into the bloodstream, circulatory system. So you can see that in a little bit more detail here, where it's coming through that membrane, entering into the bloodstream, and then it's able to kind of hide itself within the bloodstream and travel off to distant sites. And then here on this, you can see that it's gone into a little capillary, and then it comes back out of the bloodstream in and starts a cancer then at a secondary site, which is also then known as the cancer metastasizing. The cancer can also spread via the lymphatic system. So what does the lymphatic system do? Well, the lymphatic system is a really important part of our immune system. It helps protect from infection and disease, and lymph nodes are a part of this system, and lymph nodes help to filter out harmful cells from the lymph fluid. So an example of this would be if people have ever experienced a very bad sore throat, you can find glands being swollen in your neck, which would be your lymph nodes. And what's happening is there's kind of a sampling going on or a filtering, and these bacterial cells, for instance, it's causing your throat infection, are in your lymph node. And then there's very specialized cells within your lymph node that create antibodies and other immune mediators to help fight the infection and then resolve, say, for instance, your throat infection. And here you can see there's many lymph nodes around the body, and also a lot of lymph nodes around the lungs as well. So what can happen is the tumor cell can come from the primary tumor within the lung. It can break off and it can go into these lymph nodes. And then once it's in the lymph node, then it can spread around through that lymphatic system. So you can see it's pretty extensive system throughout the body. So cancers can also spread via the lymphatic system. So it's always important to remember that a lung cancer, so a primary lung cancer that starts out in the lung, that spreads to another part of the body, so that metastatic site or a secondary site, as some people refer to, it's still a lung cancer. So here again, we have our cancer cells here in sort of like a purpley color. We get that break off from the primary tumor. It goes into the bloodstream. These are my version of red blood cells here within this, within our circulatory system. And here is just an example of one of those cells that is broken off, and we were able to identify it here. And this is a study done in our own lab. So this is one of the cancer cells that's broken off for a primary tumor, and we were able to pick it up in the blood. So of course, once these cells are out into the bloodstream, they can spread around the network, as you've seen in the other two slides, and can go to places like the brain, adrenal glands, bone, and so on. So this is quite a complex process as to how cancer cells spread around the body. And to be honest, it's still not completely clear exactly how this process happens. And there's a lot of research going on into that space at the moment. And some of the things that have been identified is possibly there's an issue around the boundary of the cells. The cells can sort of disguise themselves in a way because obviously the immune system is not picking them up within the bloodstream. It could be that they change the surface of the cells so they become more sticky. And then also you can have what's been shown in some studies is there's some kind of, for want of a better word, kind of like a homing device within the cell, and it knows where it should travel to depending on where the primary tumor is. There's also a lot of work showing that platelets, so if you've ever had a cut and you develop a scab on the cut, that's through platelets and through a system that they're involved in. And there's data showing that these platelets can surround the tumor cell that's in the blood, and then that hides it then from immune destruction. So this is just an overview of some of just what we've covered in the kind of Back to Basics 101. So I'll let you have that as a reference point to go back to, but it's just really a summary of all that we've just gone through there. So the next thing that I want to talk about is around how we diagnose lung cancer. So how do we get that accurate diagnosis of such a complex disease? So we know that lung cancer is not just one disease. There's many different types of lung cancer. And as we find out more and more about the disease, it's adding more and more layers of complexity to it. So pathology helps to determine the type of lung cancer. So pathology is a term that many people would have heard quite a number of times, but basically the primary function of a pathologist is to try and help determine the type of lung cancer. And there's many different types of pathologists out there, and they do different things, whether it might be looking at molecular markers or looking at individual specific tumor types. So if we think about lung cancer specifically, well, the broad diagnosis would be around non-small cell lung cancer or small cell lung cancer. And we know within non-small cell, the three main subgroups are large cell squamous cell carcinoma and adenocarcinoma, but other types within non-small cell exist. So the histological diagnosis that's made by a pathologist is made using what's called various immunochemistry markers, or IHC. And here's just a list of some of the markers that are used in order to determine whether something's a non-small cell adeno, for instance, or a small cell lung cancer. So when we look at immunohistochemistry, I just wanted to give you some images so that you could visualize it a little bit clearer, hopefully. So here on this panel of images, this is tissue from a lung cancer, and it's put into these special blocks. So they're paraffin blocks, so it's like a wax block. So you can see the tumor here. They're cut with very special equipment. So it's kind of like you get like very thin, fine slices of the tumor so that you can look at it under a microscope. These are then stained up with special stains using the staining machines. So slides go in there, and there's different antibodies that can be used to stain up the slides. And then the pathologist will look at it down the microscope and be able to determine what type of lung cancer it is, whether it's, say, adeno or squam. So here I just wanted to give two examples. So again, just to help you visualize, for instance, here's an adenocarcinoma diagnosis where they looked at the TTF1 marker, and here's a squamous cell carcinoma diagnosis where they've looked at P63 as marker. So this brown sort of color that you can see here and here indicates that that tumor tissue is positive for that marker. So when you see that positivity, with that brown stain, it then helps to determine the diagnosis. And we also have other slides which are H&E, and a H&E is just a very generalized stain so that the pathologist can see the structure and the different types of cells within the tumor sample as well. So we've covered more of the histological markers, but we know that there's more to it than just histological markers. We know that there's also different molecular markers. So terminology is interchange between biomarker, molecular markers. So for instance, EGFR, ALK, ROS1, but of course we know there's quite a large pie chart of lots of different other alterations that can be identified within the tumor itself. And then also, of course, we have the immunomarkers as well. So one that people are probably most familiar with is something like PD-L1. So when we think about the histological diagnosis and we think about the more sort of biomarker molecular testing, this helps to get a more accurate diagnosis of the disease, which then helps to determine the right treatment option for the right person, because we know lung cancers are so different depending on the individual. So of course, when we talk about those kind of molecular markers, sort of the EGFR, ALK, ROS, and so on, there's different ways in which these can be identified. And we know that there's been great advances in the technologies that's been used. And as the technologies, the sequencing technologies continue to improve, it means we're finding out more about the disease and we're able to provide a more accurate diagnosis as well. So here's just an example of something called fluorescent in situ hybridization, or FISH for short. And basically in this example that I'm showing here, it's looking for an ALK fusion. So what they can do is they can add these probes that attach on to say ALK or EML4 in this example, and you can see that they're stained up by different colors. And then if there's a fusion present, then you can see here there is a fusion present because of that different color. So here, this is an example of an ALK fusion detected using this technique called FISH. And then when you hear a lot about sequencing, a lot of people are wondering what does that look like? Well, again, it's very big machines that do a lot of this sequencing work. But at the end, when it's sequenced, you get trace files. So here you can see the different bases that make up our DNA. And then what they're looking for along these files is to see if there's any changes. So in this example that I'm showing here in this trace file, there was an EGFR mutation present that was picked up from the sequencing. So again, just some images to try and help you visualize what this looks like within the lab. Of course, there's other tests that can be done to detect protein expression. So we've often heard the term PD-L1 overexpression. And again, the tests that are used to detect protein expression can also suggest treatment options. So again, I'm just giving one example here for PD-L1. So again, we have the H&E, which is just showing the general structure. And here we have an antibody directed to PD-L1. So again, that kind of brown stain that you're seeing there is indicative of PD-L1 positivity. We also, of course, know that not all molecular alterations are the same, nor can they be detected with the same tests. And it can really depend on where you are, and in some instances where you are treated, as to what tests might even be available. But some of the changes that can be detected, and again, these are just some of the examples, is mutation. So for instance, where you have a mutation when there's a change in only one place in the gene. So for example, KRAS, and a lot of talk has been on KRAS G12C. So where there's an alteration within that specific location within the gene. Then you can have other changes. So when we talk about genes, we know that they're made up of introns and exons. And from, for instance, here in this example of EGFR, we know that there can be mutations in terms of insertions or deletions. So again, here we have an example where we see exon 20, and then this little purple box indicates that there was an insertion mutation that went into exon 20. So you may have heard exon 20 insertion mutation. And then in this other example, we have a deletion whereby an exon has been deleted. And here in this example, it's an exon 19 deletion. Again, some terminology that you may have heard. We also know that genes can be amplified. So MET is an example of that. So you should have your, so here again, my kind of little blue box, I guess, with
Hello.
MET. So, oh, I can
Hi,
hear someone in the
Seth. How are
background.
you?
Good,
Thank you.
I'm really sorry, someone's talking in the background.
Okay,
If they're
but how
able to
long?
mute. So you can get something that happens, again, kind of like the examples I showed with the mutations, and you get then what's called gene amplification. So then you've multiple copies of the MET gene. I've also touched upon the fusions when I showed you the fish example. So here we have EML4 and we've ALK. Something has happened, there's a fusion. So you get the fusion of a number of exons from EML4 and ALK coming together for that fusion. And then I did touch upon this a little bit about that protein overexpression. So again, you're going from, say, for instance, what this would be representing as normal protein expression to something where something's gone wrong then, and you have the overexpression there on this cell. So getting the histological diagnosis and getting the sort of biomarker molecular testing information is just one part of the process, because obviously staging helps also to determine treatment options, staging and obviously how you feel yourself and what your treatment goals may be. So very briefly, I'm just going to go through this. So if we look at non-small cell lung cancer, it's staged based on the TNM. So T is indicative of the tumor, N is in relation to the lymph nodes, and M is related to whether the tumor has spread. So T describes the size of the tumor, N describes if there are any cancer cells in the lymph nodes, and M then is describing whether there's been spread to a different part of the body. So, and again, I should reiterate that this is a very generalized, brief overview of staging. It's more complex than this, and it can be further divided. But in general, you have stage 1, where the cancer is small and hasn't spread, stage 2, where it has spread a little bit from where it started. Both of these would really be termed early. Stage 3, where there's spread to the lymph nodes and nearby tissue. And then you can have sort of stage 4A, 4B, and this is around the spread, whether it's to a limited number of sites or spread to several areas in one or more organs. And then a term you may have come across is oligometastatic. So that's kind of like an intermediary stage of non-small cell, and it's sort of between localized and widely spread disease, and it's characterized by a limited number of metastases. So as you can see, at each stage of the process here, there is a lot of complexity when it comes to lung cancer. So again, this is just a short summary slide, just showing the different things that we spoke about in our Back to Basics around the diagnosis of lung cancer, both the histological diagnosis and also the molecular biomarker alteration testing as well. So where are we? We can see that we have made advancements in our knowledge in the biology and all of that within the space. So what has that meant? Well, here's the most recent data from SEER. in the US, where we see where survival rates, 5-year survival rate was much lower. If we look into the 70s versus where we are now, we can see that the 5-year survival rate for lung cancer is improving. And the most recent data from SEER covers the 2014 to 2020, where 5-year relative survival is 26.7%. So we're going from here to there. And of course, hopefully what we're going to continue to see is that 5-year survival rate for lung cancer improving. And a lot of that is down to the research that's been undertaken to not only understand the biology, but also to find new treatments as well for the disease. So some of the terminology or some of the different treatment options that you may have availed of or know someone else might have had. You might know someone who's maybe only had one form of treatment, or you might know someone who's had a combination of different treatments. So this is going to vary on exactly what stage of disease your tumor is, and also all of those other things we talked about, the biomarkers, the histological diagnosis, and all of those types of things. But surgery may be an option, chemotherapy, radiotherapy, targeted therapy, immunotherapy. It's really just going to depend on your own situation. And then some of the terminologies that you may hear around cancer treatment is things like combination therapy, so 2 or more drugs given together, doublet chemotherapy, so it's 2 chemotherapies, or you may have also heard the term adjuvant, which means something that's given after surgery, or neoadjuvant, which is something given before surgery. So there are just some of the terminologies you may have come across. So on average, when we look at the different changes within lung cancers, within lung cancer tumors, and that's what somatic mutation refers to. Somatic mutation is in relation to mutations that happen within the tumor. We can see from this graph here where you've lung adenocarcinoma and lung squamous carcinoma, that compared to a number of other cancer types, there's a greater number of mutations. So on average, lung cancers have more somatic mutations than other cancer types, and that's just what's being demonstrated here by this figure. Oh, and we also know with the more that we're finding out about the lung cancer, the discovery of new treatments is also increasing rapidly. So this is from a paper published in 2023, but we know the situation is changing all of the time. And in some years, there's been such rapid change that it's been a little bit difficult to keep up. But you can see there's differences between the first line and second line setting, and you can see just some of the different drugs and sort of the timeline of when they came online. Now, this slide is a relatively old slide, but it is one of the best slides that I can find to illustrate the point that there have been a lot of new drugs made available since 2008, but I should say this slide is out of date. It's here for illustrative purposes. But this chart is from 2015 and it's already out of date because we know there's lots of newer treatments and other markers that have come on stream since then. So again, it's underscoring that improvement that we're having within this space. Oh, and I was going to just mention, obviously, a lot of discussion has been about KRAS in more recent times, but we still know there's a number of unknown oncogene driver mutations as well in a significant cohort of people. So this slide, I know it's very busy, but it's just to show again that the landscape is becoming very increasingly complex. So you can see the different types, whether it's immuno-oncology or non-immuno-oncology related, so the targeted therapies, the chemotherapies, and the different things that they might be targeting. And then with the immuno-oncology space as well, you can see that there's a number of targets here, signaling, vaccines, all sorts of things going on. So it is becoming complex. And here on this side, we can just see a number of kind of targets that are either in a preclinical or different phases within the trials for non-small cell and small cell lung cancer. So I'm just going to touch upon immunotherapy, but basically immunotherapy is something that helps the immune system to kill cancer cells. So this example is showing a white blood cell gobbling up a bacteria because it's recognized as foreign, but basically the premises of immunotherapy is to help your white blood cells recognize tumor cells and help to kill the cancer cells. And here again is just a very brief overview of the number of immunotherapy approvals for lung cancer and the different things that are happening within that space. And again, there's been more happening since this was published in 2022. It is important to note, though, that there are other immunotherapies other than just immune checkpoint inhibitors, that there's a lot of research going on. And obviously there's other treatments options out there as well, like antibody drug conjugates and so on. So this is just a brief summary of where we are. But of course, the most important thing is that survival is improving. So what might be next? Well, I think all of us have heard a lot about AI, machine learning. I think there's going to be a lot still happening within this space in terms of screening, diagnostics, and treatment decision. We can see it being mentioned time and time again. We also know within the diagnostic space, we're still hearing a lot about liquid biopsy to help with screening, single cell analysis, microbiome, a lot of different things going on within that space, and to try and make the diagnostics less invasive, but to help with the accuracy of the diagnosis as well. And then within the treatment space, there's been a lot of movement with early stage disease. There's been movement in treatment sequencing and combination, new drugs for new targets, drug resistance, and tumor vaccines. And of course, with clinical trials, I think there's still more to go to ensure that we've real world patient inclusion and access. So hopefully, lung cancer will become more of a chronic disease with all of these different changes and improvements that we're seeing. And with that, I'll end and I'll stop sharing.
Thank you, Anne Marie. That's always a lot of information to digest.
A lot.
So we have about 15 minutes left in the session, and I've got about a dozen questions to cover. So we'll see what we can get through.
Yeah.
First of all, there was a question about what is an exon?
An exon. So an exon was traditionally found to be something that wasn't gene coding. So it was something that was referred to as like junk DNA. But we know now with much more research that's gone in that there's things within these exons that help with that regulation of gene expression. And there's a number of groups working on that at the moment, because I know I showed it very simplistically, but obviously that regulation is much more complex than that.
Right. So is there a common characteristic of all forms of lung cancer?
There's not really a common characteristic as such, because we know that lung cancers are so different. It's really, it's that it arises within the lung cells and that it has a mutational profile. And a lot of lung cancers cause the same kind of symptoms, but really we know that it is very different depending on the type of lung cancer that is diagnosed. So really there's not huge commonalities within it because they're all so different.
It's pretty much the only thing that's in common is that it started in cells in the lung.
Yeah, basically. And you can see how complex it is because there's so many different cell types. So
right. So can the biology of lung cancer give us any insight into what caused a particular case of lung cancer?
So the very short answer to this would be yes. You can see that, if there was a smoking history, you can kind of see like kind of areas that would be modified. So sometimes referred to sort of as fingerprints that can be indicated of sort of more smoking related damage. There's some evidence to suggest that where there might be a viral influence and picking that up. But I mean, the data is a bit conflicting there. And there's things like if you've been exposed to asbestos, for instance, you can find asbestos bodies within the lung as well. So there definitely are things that can help give insight into what might have been one of the risk factors that were at play.
Can diet or nutrition influence cell mutation?
So with diet, there are studies out there, but again, it is a bit conflicting when it comes to the lung cancer space. There's study that shows that diet has an impact. There's studies that showing diet doesn't have an impact. But what I would say in general is there can sometimes be instances of deficiencies. So for example, vitamin B deficiency, whereby then it impacts the DNA synthesis. So if you don't have that B vitamins there, it can impact that synthesis, and then you can get genomic instability. Something that is being looked at a lot with diet is around epigenetics. So that's not mutations within your DNA, it's just the structure of the DNA as to what genes can be accessed to be switched on and off. And there's studies showing that diet can really impact that. But as for diet having a direct link on lung cancer risk, the data is inconclusive.
So you've talked a lot about the genetic influences. Excuse me. How about some of the studies that show cancer might be a metabolic disease? Can you talk about how metabolism might influence the development of cancer?
So there's a number of different theories around the kind of metabolomics of it, and something that came up quite a long time ago was something called the Warburg effect. And what that indicated was that tumor cells switch their energy metabolism. So they switch different pathways. And then when they switch different pathways as to how they make their energy, obviously the end products are then different as well, which can impact it. There's also studies that feel that it's all related to what goes on within the mitochondria. So the mitochondria is also known as the powerhouse of the cell. So it's providing all the energy. But there's studies showing that there's differences happening within there that again is related to energy metabolism and those products that are made. And then that can influence the cancer as well. So there's a lot going on within the space around metabolomics and energy metabolism.
Okay, so if someone has their lung cancer tested for mutations and no mutations are found, does that mean the cell just doesn't have any mutations at all?
No, it means there's still mutations. We just haven't figured out what mutations might be there, because it's not going to occur without that stepwise process of accumulating mutations, particularly within those genes we talked about, the tumor suppressor genes and the proto-oncogenes. If we look at lung cancer, somewhere in the region between 50 and 60% has a mutation or an alteration within that P53 that I spoke about, that tumor suppressor gene. So that's one of the very common ones, but we don't have a targeted therapy for that. And I think the other thing to understand is when tumors are sent for testing, you know, it's usually just the more known or more common markers that are looked for, sort of the EGFR, the ROS1 and so on. But there's definitely mutations and changes is there. We just haven't identified them.
And sometimes you'll get your test results back and it says there is a mutation of unknown significance. The gene doesn't look like what they expect it to look like, but they have no idea what it means.
Yeah. And that's going to happen as our sequencing technologies improve, as places are doing more broader sequencing panels. Chances are we are going to find them, like for instance, in breast and other cancers, there's a number of changes that they've identified, but they're still not really sure what it means. So we just need to build on the data and do those larger studies so then that we can say with I can't think of a better word, authority, which is not the word that I'm looking for, but whether or not it does have an impact in the development or the progression of lung cancer.
So somebody has lung cancer and they're being treated for it, and then they have a progression. Can they acquire a new mutation, or can there be more than one mutation in a cancer cell?
So you can have co-occurring mutations, because when you think about it, back to that slide of that really high somatic tumor burden that we see within lung cancer, there can be more than one mutation. And then if you say, for example, have, and again, I'm just using this as an example, I know I keep picking on EGFR, but for instance, if you've an EGFR mutation and then there's treatment and then a resistance mutation develops, more than likely you will have retained that initial mutation as well as the resistance mutation. And there's models that kind of show why that's the case. It could be that you're selecting out with your treatment pressure. So if you think about it, very basic terms, antibiotic resistance, it's something kind of along the same vein. You can also get what's called persister cells. So even before you treat, there's just cells there that are that little bit different because of the heterogeneity within your cells. And then you can have cells that one could have an EGFR mutation, but the other cell might not have an EGFR mutation, but then there could be something else. So again, it's quite complex because we see that heterogeneity.
Are there some populations or people that are more likely to get lung cancer? For instance, we see a lot of Asian women developing EGFR positive lung cancer.
Yeah, so there is definitely an ethnic relationship in terms of lung cancer risk. And if some people might have heard of something called the Talent study that was done in Taiwan, and it looked at a number of risk factors outside of tobacco history and tobacco use. And they found within their population, there was issues around absence of ventilation during cooking. They had a thing called the cooking index, because if you don't have ventilation when you're cooking and you're using oils at very high temperatures, they can release volatile compounds and you're breathing that in. I mean, there's exposure to passive smoking as well. You could have maybe something like COPD, TB, or another inflammatory condition, and all of those can impact. And they're definitely seen in more Asian populations. It's to do with some of these risk factors around that exposure with the environment. Because the vast majority, I think in Taiwan, where the Talent study was done, there was no smoking history within the general population of those that got lung cancer.
Do family members of a lung cancer survivor have a higher risk of getting lung cancer?
So, well, in that Talent study, yes, it showed that within that non-smoking population, that a first-degree family member was indicative of a risk. But if you think about it in general, a family living together, they're exposed to similar things. So whether it's something like that, or maybe radon or asbestos or whatever it might be, they're also getting exposed to those risk factors. So that is something to be conscious of. There is some data out there showing that there's germline mutations. So germline mutations linked to lung cancer. So a germline mutation is something within your DNA that's hereditary that can be passed on. So there's some data indicating that there can be a link there for some people that have lung cancer, and that's mostly around EGFR where the data is on that. But germline testing for lung cancer is not something that's standardly done, but we know there's a lot of research going into that area at the moment as well.
So are we really seeing a greater increase of lung cancer in certain populations, such as young women, or could that just be because we're measuring more people, or there have been some lifestyle changes?
That's a great question. And I think depending on who you ask, you're going to get a different answer. But I think it's a lot to do with the environment and the exposures that we find ourselves dealing with. There's a lot of environmental pollutants that people are exposed to on a daily basis. There's evidence suggesting that females in particular are more sensitive to those pollutants. There's also some evidence showing that you have these genes that make what's called detoxifying enzymes. So say you're exposed to something that could cause a problem in your body, you should be able to deal with that to kind of eliminate it so it doesn't cause a problem. But there's data showing that in certain females, there's an issue with sort of detoxifying that thing that's coming in to cause you harm. And then over time, getting exposure to that is going to, increase that risk, that likelihood of developing the disease. So for me, I think it's a lot to do with environment. And I think in females as well, there's definitely that hormone issue, like the sex hormone issue. And I know there's people researching into that at the moment as well, because we know that females who might smoke don't have to smoke as long or as much as a male in order for their risk to increase. So there's obviously something there in the background, but we still haven't fully figured that out.
Well, thank you for all of your knowledge and sharing it with us and answering the questions. There was one question and within 3 about, is there a place if you want to learn more about how the cell works? And I think a really good place to start is a book called The Song of the Cell. It's written by the same man who wrote The Emperor of All Maladies about cancer, Siddhartha Mukherjee. And it gives you a really good overview of all the things going on in the cell, which is an incredibly busy place.
It is.
Thank you so much, Anne Marie, and thank you all for attending, and we'll see you next month.